Hello and welcome to Lecture 22, Epigenetics and Regulation of Gene Expression. So let me start today by having us think about why we would want to regulate gene expression. So essentially we have the genes in the genome, but they're not always on, they're not always active. Not all of them are active. And there's a very elaborate way of telling the cell, or for the cell to sense when genes should be expressed. And to understand why it has to be so difficult, so complicated, so elaborate, it makes good sense to think about why that even makes sense for the cell to have that kind of a system. And that really is because there are so many genes, and most of them are not needed most of the time. Most of these genes only are important for a certain period of time, for example, during certain developmental phases, when the organism is very small, when it's maturing, when it's old, but not at all times. And then there are a number of genes that are responding to the environment. So, for example, when the organism takes up food, then there is a reason for certain enzymes to digest this food, which is not given when there isn't that particular kind of food around. So it makes good sense that the cell can sense what is required of it to do at a given time, and then use that input and determine which genes need to be on to best respond to this particular stimulus. So that will, of course, help the cell then to conserve resources, right? So it's not having everything on at the same time. It doesn't have to produce a lot of protein at the same time that it doesn't need, and it conserves the resources. So chapter 19 in your book looks at all kinds of regulation, of gene regulation, at a variety of different types of cells and aspects of life. And so there's a lot of information, but the gist of this chapter is really for you to see the diversity of different kinds of regulation, both in prokaryotes, bacteria, for example, and in eukaryotes. So we're going to start here with the eukaryotic genes and how they regulate the cell. And it's an overview, and then we're going to go through all of these individual steps, so step by step later on in the lecture. Let me start by this picture here, figure 19.1 from your book, that shows that DNA is wrapped around histones, so those are proteins, and these proteins allow the DNA to wrap around something, and then that can be compacted so that it becomes a really dense molecule that's easy to move, for example, during mitosis or meiosis. Right? So you don't have to yank these spaghetti thin strands. You have something that's more compact, like these chromosomes that you can see in the microscope. And in order to get that, you get compaction because you get this tight wrapping of DNA around the nucleosomes. But it also helps, even when the DNA is not fully compacted, like during cell division, it's usually compacted to some degree. And that helps, again, with it just not being tangled and it being a complete mess in the cell. Now that means that if transcription is to happen, then the enzymes, the machinery of the cell that transcribe the cell, that transcribe it and then have the RNA be translated, that they have to gain access to the DNA and to the particular genes that need to be expressed. And so if they're all bunched up like this, then they can't really get those enzymes can't really get to the DNA that needs to be transcribed. And so these nucleosomes have to be moved around. Some of these proteins have to be displaced a little bit so that there's access, that the other proteins that are needed for transcription can gain that access to the DNA. And so we'll talk about chromatin compaction, and we could talk about histone modification. These are the histones again. Together with the DNA, they make the nucleosomes. We talk about how these are modified in order for this transcriptional machinery to recognize what genes should be active and what should be passive. Then we have transcription, of course, of the RNA, and then there's processing of the RNA. And we've talked about exon and intron splicing. We've talked about alternative splicing. We've talked about the the capping of the 5' end and the 3' end with a poly A tail and the 5' cap for stability reasons of the RNA, which is then translated in the cytosol or the cytoplasm, and the ribosomes that then make the proteins. They have to find the mRNA, and they have to find the starting point, and they have to be recruited to the RNA, and that all is also regulated when ribosomes will bind to RNA. And so we have transcriptional regulation, but we also have post-transcriptional regulation after it was transcribed. And then we have translational regulation, which is during the time when it's translated by these ribosomes. And then even after that, when the protein is ready to be used, we can still modify it by adding certain groups, chemical groups like phosphate groups or methyl groups or acetyl groups onto this protein, and that is then referred to as post-translational regulation. So we have transcriptional regulation, chromatin compaction, histone modification, these general and regulatory transcription factors. These are proteins that are allowing the recruitment of the RNA polymerase to the DNA to start the transcription. And we have DNA methylation. So that is another aspect of regulating the DNA during or before transcription. And then we have the post-transcriptional regulation through splicing and processing, in a process that's called editing that you haven't heard about. So we'll talk about that a little bit. And then there's the translational regulation, where RNA binding proteins facilitate the translational initiation and termination. So certain proteins need to bind here before the ribosome can assemble. And then there's translational inhibition via a RNA-induced silencing complex. We'll talk a little bit about that as well. It's called the RISC, or the RISC complex, even though that C stands for complex already. We have RNA degradation as a way to regulate whether or not protein should be made. And we have induction of more chromatin remodeling. So in other words, chromatin remodeling, which happens over here during or before transcription, some of the RNAs that are made can again regulate, sort of in a feedback regulation, the transcription of the genes themselves. We'll talk about that as well. And then there's the post-translational regulation, where you have the modification with the chemical groups. So all of this is going to be topic of today's lecture. Some of it you should already recognize because we discussed it early on in the semester. Other things we haven't discussed, and we'll focus on those today. Okay, so histone modifications are a way for DNA to become more or less accessible to the RNA polymerase. And the way that histone modifications are regulated, in other words, how transcription is regulated on the transcriptional level or even the pre-transcriptional level, is by using these, they're called histone tails. Those are the ends of the histones. Now, the way that this figure shows this is there are 8 proteins. You see that they sort of like an octamer here, right? This is 1, this is 2, 3, 4, and then here's another 4 on the bottom. And these little green squares in here, those are the amino acids. So each of the red boxes is a chain of amino acids. It's a protein, right? So it has lots of amino acids that are sort of bunched up together in some 3-dimensional structure. But all of these histone proteins have a tail, they have an end, and that sticks out. That's not coiled up into the rest of the protein. And these histone tails, these ends of the histones, those are individual amino acids. Of course, again, this is the first one from the end, the second one from the end, the third one from the end, the fourth one, and so forth and so forth. And specific amino acids that this tail is made up of attract certain kind of modifications. Oftentimes lysines, that's one of the amino acids, if you remember, lysines are methylated, and when they are methylated, when there's an extra methyl group stuck onto the lysine, either 1 or 2 or 3 methyl groups, in this case it's 3 methylated, they can have an effect on the activity of the gene, the blue strand here, that is wrapped around the histone, which is modified. So the DNA that's in this blue strand, right? That is the DNA. at this particular location in the genome where the histones around which that DNA is wrapped are modified or not modified, are either transcriptionally active or passive, depending on the modification of the histones around which they are wrapped. So we have a number of groups, as I said, the methyl and the acetyl groups. Methyl groups can activate or deactivate transcription. That depends on their location. So they're not all here on this particular amino acid that your book suggests, but they could be over here on the 4th or the 9th or the 27th amino acid. And each location of the amino acid means something different for the activity of the gene. And having 1 or having 3 may also be the difference between being active or being passive. With acetyl groups, it's usually so that they activate gene transcription. Here's an acetyl group, right? This is the acetyl, almost like acetic acid. That's where the term, of course, comes from. Those usually activate transcription. I say it's important to realize histones are proteins made up of amino acids that are coiled up and form octameric complexes around which the DNA is wrapped. So that is histone modification, and that type of modification determines whether or not a gene is active or passive. Or at least it helps with that. It's not the only thing that determines it, of course. Another way of regulating transcription is another type of modification, but this time we're not modifying the histones. This time we're modifying the DNA itself. And so here what we have is a modification of the DNA of the nucleotides with a methyl group. So your book has this figure, and it shows an undermethylated region and a heavily methylated region. And so under means less, obviously less methylated, and heavily methylated, and tons of methylation. And you can see these little groups here indicated with the yellow circles and the reds, with the red circle around the yellow dot. This entire region here is heavily methylated. Now, let's first speak about what methylation is. So if you have a cytosine, of course, that's one of those 4 nucleotides, the C, ATCG, that's the C, and the C is often methylated. And when it's not methylated, it looks like this. And when it's methylated, there's a methyl group, CH3, that's the methyl group, that is attached here to the ring of the methyl group, of the cytosine, and it becomes 5-methylcytosine. So each of these little yellow strands now are methylated cytosines, and they often occur in clusters. And these clusters are referred to as CPG islands. Kind of a dumb name because it's confusing. Why is there this P in there? The P really stands for the phosphodiester in between a C and a G. You could just call it a CG island, or an area that is heavily enriched in methylcytosine. But often in the literature you will find this CPG island, or CNG islands as well, CNG regions where the N could be another nucleotide in there that is not defined. N is in any of the 4. So the point here is that when you have methylation in a region, especially when it's a promoter and it sits in front of the transcriptional unit, so the part of the DNA that is transcribed, then transcription oftentimes is blocked. So you can have an addition of methylation in the promoter region of a gene which will block transcription. And when it's not there, then the undermethylated region will not prevent transcription, and so transcription can occur. So that's another way how genes can be regulated through the modification of DNA. Now, this slide, the title of this chapter really says epigenetics, and epigenetics means epi means on top, genetics means the genes on top of the genes. These 2 examples that I just talked about, the DNA methylation, that is an example of epigenetics, because it's not the genes per se that are changed. It's not the DNA sequence that's changed. It's still a cytosine. It's still a thymine and adenine and so forth. but on top of it is another layer of regulation. That's that methylation. So that's why it's called epigenetics. And that's also true for the histone modifications. Again, that's another layer on top of the DNA, or underneath the DNA, if you will, because the DNA is wrapped around these histones. And that's why that is also referred to as epigenetic regulation. So we have two types of methods here for epigenetic regulation. Okay, so an example of why epigenetic regulation is really important is the example of the X inactivation in mammals. So in females of mammals. Remember that females have 2 X chromosomes, males only have 1 X chromosome, and if genes are expressed twice as strongly, those that are on the X-axis, on the X chromosome, then they would be in the males, for example. Then there could be a difference in the way that these genes act, the way that they respond, the way that they function, and so usually 1 X chromosome shortly after conception, after the formation of the zygote. But not immediately during the formation of the zygote, but a little bit after that, is inactivated, one of the two, randomly, so that females also functionally during their lifetime really only have 1 X chromosome, making the same amount of product from their single active X chromosome as the male does from his single only X chromosome. And the way that this works is by a gene that's called XIST, and it sits on the X chromosome, and it makes an RNA. The XIST gene is transcribed and spliced, and it's a non-coding RNA. So it's an RNA product that doesn't code for protein. It's not the protein that does the job. It's the RNA itself that does the job in this case. So it binds with the X chromosome inactivation center. That's where it starts. And then it spreads along the entire length of the chromosome. And the presence of this XIST RNA triggers DNA methylation. So after all this RNA is there, it sort of codes the DNA. Then DNA is methylated. So that's what we just talked about before, right? Cytosine methylation, which then reduces the transcriptional activity. So I say here RNA molecules transcribed from a specific locus, spreads across the X chromosome. The presence of this special RNA recruits enzymes that transfer methyl groups to the DNA, and then lots of these methyl groups make the chromosome bunched up and tightly wound, inaccessible for transcription. And so that is also sometimes referred as transcriptional silencing. So the X chromosome is inactivated or it's silenced. And here's an interesting example of what you can see, or how you can see this phenomenon of X inactivation in a mammal, in a female mammal. There's a particular gene on X chromosomes in cats that make this calico type of patterning on cats. And the reason why you have that is because you start with a maternal cell that has an X and a Y. I mean, an X and from the maternal and an X from the paternal donor in the fertilized egg. And as I just said earlier, one of these X chromosomes is inactivated, but not immediately after the zygote formation, after the fertilized egg formation. So it goes through a number of cell divisions, and then at that point, one of them becomes inactive. So that's when the RNA, this XIST RNA, is transcribed and coats the chromosome, and then the chromosome becomes methylated and epigenetics silenced. That means that from then on, this cell will give rise, every time it divides, to a sector, a patch of this female that has in this case, the paternal chromosome silenced and the paternal active. But in a different patch, in a different part of the body of this female, a different chromosome is active and the other one silenced. And each of those will now give rise to a bunch of other cells that come after it throughout the development of this female. And so you get sectors, and these sectors are sometimes referred to as mosaics. A mosaic is a piece of entirety that's put together out of different sections. So you have random X inactivation in the embryo. It occurs about the time of the implantation in the uterine wall. And in each cell lineage, this inactivated X remains inactive for the rest of the life. And that is why in cats, which have a gene for coat color on the X chromosome, where either one or the other can be silenced, they show this patchy coat color because each of the patches refers to one kind of X chromosome that's active or not. So you see here the mosaic black and orange colors result from X inactivation. In the black, one is active, and in the orange, the other one is active. The white patches are due to the expression of a different gene. So that is that does not explain the different colors, but the X inactivation explains the black and the orange. So that's an interesting phenotype that arises from this mosaic in the females. All right, so let's get back to our main topic here, transcriptional regulation. So we talked about transcriptional regulation with respect to epigenetic modification. So you can regulate transcription through epigenetics, and there we had the histone modifications, and we had the DNA methylation. And now we're looking at transcriptional regulation with transcription factors. And this should be familiar to you. It should look like something we've discussed early on. I think it was chapter 4. Where you have the DNA, the promoter of the gene that sits here, and the gene that is the coding region of the DNA for that particular protein later on that is going to be transcribed and then translated. That starts over here. So the direction of transcription is right here, where the RNA polymerase, that's this green thing here, that is made up of multiple subunits, proteins made up of multiple subunits. They bind to the promoter, and they can only bind when certain regulatory transcription factors bind to these so-called general transcription factors. So there's some transcription factors that are always necessary, they always have to be there for every gene. And then there are some transcription factors, proteins, right? Transcription factors are proteins, that only bind to specific genes. So you need to have that specific transcription factor binding to a specific gene that will turn that gene on at just that time when all the stars are aligned. In this case, meaning when all the transcription factors are aligned such that they will then recruit this RNA polymerase to start the transcription. And in addition to these general transcription factors, we also have enhancer sequences. That's these red parts here, where you have regulatory transcription factors bind as well. So they can bind over here. They bind to these other scaffolding transcription factors, general transcription factors, as well as the regulatory or specific ones. All that together will put the DNA just in the right shape and the RNA polymerase in place for the transcription of the gene to occur. So I think if you remember that there are general transcription factors and specific transcription factors, or regulatory transcription factors, that's the same thing, that will then recruit the RNA polymerase to initiate transcription. And so that is usually referred to as sort of the transcriptional regulation, not post, not pre, that is transcriptional regulation. And then after transcriptional activity, when the gene has been transcribed and RNA has been made, then multiple mechanisms of post-transcriptional regulation start to occur. And we have talked about the splicing, so I'm not going to go over that here in any detail. You can have alternative splicing, right, where you have one exon either present or active. You have a different type of selection of exons that are being expressed that are actually in the end make it into the protein sequence. In some cases, those exons are left out, and that difference between those different types is called alternative splicing. And so here's an example where you have the mRNA from this particular gene expressed in the liver in such a way that exon 11 is present, it makes a particular kind of receptor, and in the skeletal muscle, where less of that is required, of this affinity, it makes where more is required, it makes a high insulin affinity receptor. Here it makes the low insulin receptor. So it's just an example here. This is not about this particular gene. This is about alternative splicing as another way of post-transcriptional regulation. Okay, so now we come to a process that we haven't discussed before, and it's called RNA editing. And so it was only found 30 years ago or so. So transcriptional regulation was long known, but what was not known is that RNA, after it's transcribed and spliced and modified, can still be edited. And so there's a suite of proteins, and there's actually still not that much known about how this process works. It's just known that it does happen. And so what happens then is that certain enzymes can take the RNA nucleotides, the nucleotides that are part of the mRNA, and they can change them to a different chemical molecule. So adenosine can be deaminated, so that means this amine group can be removed. And when it's removed, then adenine is no longer called, or adenosine is no longer called adenosine, but inosine. Now it has this oxygen here. It looks different. That's the point. So chemicals make this nucleotide look different. And when it looks different, that has consequences for how it's translated. And the same is true for cytosine, that can become uracil, that now doesn't have this amine here, has now oxygen here, and so then that is uracil. So there's a deamination of cytosine or of adenosine. And the point is that when they are changed, then the cell machinery will look at them differently, will do different things with it, will interpret them differently. So RNA editing changes the sequence of the transcribed RNA. It does not change the sequence of the DNA, right? So this is after the fact. And let's take a look here at an example. So if you have a processed mRNA that has this particular sequence and makes this particular set of amino acids and puts them together in this particular sequence in the protein, that then has a specific function. But if this cytosine over here is edited to a uracil, it no longer codes for a C, it codes for a U. And if you look at what the codons, and what the code structure here means, if you have UAU, that stands for tyrosine, you can look that up in this code, right? In this chart of the amino acid code. AUG will produce another methionine, AUA will produce an isoleucine, and then CAA used to produce a glutamine, but UAA is a stop codon. So that means that the editing of the RNA, so this is after the fact, after the transcription, now it's edited, now it suddenly becomes a mRNA that will stop the translation here and will produce a different type of protein that has a different function. So unedited versus edited RNA of the same gene in different tissues can lead to proteins with different functions. Editing can restore wild-type protein function to mutated genes by repairing on the RNA level, right? So you could imagine that if you have a mutation over here somewhere, it could be repaired through the editing process. That does not happen very frequently, but it has been shown to happen. It's sort of a, after the fact repair mechanism, when the DNA repair mechanisms that we've discussed previously, when they don't work, when they fail, there's some sort of a stopgap possibility here for the cell to repair the RNA using RNA editing. But this is not something that usually happens. This happens in very few cases. So it has been shown for specific genes, and there it happens all the time, but it's not a general repair process. There are still large gaps in our knowledge. If you were to look for primary literature about RNA editing, of course you will find a lot, but you will also notice that it's relatively sparse compared to other areas of research that are active. DNA sequence requirement for specificity can result in allele-specific editing. So what I mean here is that this machinery of editing proteins that are doing the editing, they don't just go randomly in and edit any C to a U and then see what happens. There are specific regions in the RNA molecule that have evolved in such a way that there are proteins, or the proteins have evolved in such a way that they recognize that sequence, that they bind there, and then that allows the editing of the specific cytosine. So it's not a free-for-all where anything goes and you can just nilly-willy edit things around. Specific proteins that recruit these that are recruited to these sites, specific sites where editing occurs. So it occurs in a few places in the cell. It does not happen widely. All right, so but nonetheless, editing is another mechanism of gene regulation, right? And that's what this chapter is about. So here's another way how genes can be regulated, and that is through the use of so-called small RNAs. Small RNAs, they occur in two flavors. They actually occur in more than two flavors, but your book talks about two main flavors, and those are the small interfering RNAs, siRNAs. I think I have that somewhere written up, maybe not on this slide. Si, or small interfering RNAs, and microRNAs. So microRNAs, that's one word really. So those are not translated into protein, and instead they're used to regulate the transcription of other genes. So normally, of course, mRNAs, which these are mRNAs, right? These are RNAs, but they're not messenger RNAs that are being translated into protein. These are RNAs that are used for regulation. And so we call them si or microRNAs. So when they are made, what they all have in common is that they produce double-stranded RNA. They produce a sequence here that is somewhere along on the end, if you will, of the same molecule, complementary to itself. And it produces these hairpins, called a hairpin because it looks sort of like an old-fashioned hairpin. And of course, these boxes are the nucleotides, and they can align with each other because they are complementary. So that is what is common to these specific kind of RNAs. And interestingly, about small RNAs, or about these RNAs that are that make these hairpin structures, is that then they become double-stranded. And that double-stranded RNA is not something that a eukaryotic cell normally deals with. In fact, a eukaryotic cell has evolved to recognize mRNAs that are double-stranded as something weird, something that could be a virus. Remember that when we talked about viruses, that viruses can exist as mRNA, in mRNA, with mRNA genomes, and they can be, so RNA genomes, let's call them, and they can be double-stranded RNA viruses. And so the recognition of double-stranded RNA might be an adaptation to recognizing this weird molecule that isn't usually there as something that needs to be dealt with. And it is dealt with by an enzyme that eukaryotic cells have that cut this particular DNA. It's called the dicer enzyme. That's not in your book. It's also not so important that we need to write it down. It dices and slices and cuts this DNA into these small pieces. And that is what all of these small interfering or microRNAs have in common. Something processes them. That's what it says. Precursors are extensively processed into these small 20 to 25 base pair nucleotide fragments that are still double-stranded. So they get processed because they are recognized as something odd, and over time, this oddness, this enzyme that has made this short work of the double-stranded pieces into these smaller pieces has evolved not only as a recognition system for potential virus invasion, but now the cells have actually adopted as a way to regulate their own genes. So Now some of their own genes make these double-stranded RNAs in hairprints. It's not just invading RNA from viruses. So now there are some cells, there are some genes in cells that make these double-stranded RNAs. In fact, quite a few. There's hundreds, maybe thousands of those around. It's a whole layer of regulation that has only been known to scientists for the last 10 to 15 years. Once these small processed pieces are available, they get loaded onto this set of proteins, and it's called the RISC complex, or the RNA, now I can't remember, they're small RISC, the RISC complex, small regulatory RNAs that are then incorporated into this complex. And the complex binds the RNA. It takes the other strand off, and now it is basically a search beacon, right? It can now use this to find that kind of sequence elsewhere in the mRNA pool of the cell. So any RNA that has the opposite of this sequence can be recognized, can be specifically recognized, and these enzymes from the RISC complex that are bound with this search search mechanism can then bind to where it fits and can either inhibit the translation of this particular mRNA, so that's translational inhibition. It can even lead to the fact that it just sits there long enough until the cell says, well, this has been here long enough, let's get rid of it, and the RNA is degraded. And it can take this mRNA as a way to look for DNA of this sequence and then have this protein complex bind back to the DNA. So this would go back into the cell, past the RNA stage, into the DNA stage somewhere, and then bind on the DNA, recruit methylases, and then have that DNA methylated. And you know what happens when DNA is methylated. We talked about that at the beginning. That will lead to silencing of the DNA. So it can lead to chromatin remodeling, and chromatin that is remodeled to be heavily methylated is transcriptionally silent. So it can use that to basically silence its own genes and make sure that they stay silent by this inhibitory feedback mechanism. So that's a lot of information. I think what's important for you to remember is over here on the side, certain DNA sequences give rise to double-stranded DNA. Since double-stranded RNA is unusual in the cell, it triggers a specific response. An enzyme recognizes the double-stranded RNA, cuts it into small pieces, loads it onto the protein complex called RISC. And that RNA loaded onto RISC scans the genome for complementary regions. When it finds a complement, it prevents translation, or it causes DNA to be methylated if it's DNA that it finds. So those are our microRNAs. MicroRNAs are those that are dealing with RNA, and small interfering RNAs are the ones that eventually usually lead to the transcriptional silencing through DNA methylation. All right, so now let's look at translational regulation. What we had before was transcriptional regulation or post-transcriptional regulation. Now we're looking at how gene regulation is regulated on the translational level. So here you see an mRNA, and it has an ORF, an open reading frame. That's the coding region of the DNA. This will become protein later on. And then we have the untranslated regions. We talked about that earlier on in the class. The 3' untranslated region with the poly A tail, 5' untranslated with the 5' cap. All your book is adding to what you already know in this chapter is that there are certain regions in the mRNA that are required for translational regulation and certain mechanisms. And the regions that are important that tell proteins that are regulating the recruitment of the ribosome, right? The ribosome is what then scans around here and looks, reads the code and puts in the amino acid chain that grows. So the regions are the untranslated regions, these two here, the 5' cap and the poly A tail. That's where proteins bind and initiate eventually translation. And then the mechanism is through the regulatory regions that I mentioned here, we have the recruitment of proteins that bind, and once all those proteins are assembled correctly, then the translation process starts. So if we're talking about gene regulation, then one way to regulate the transcriptional activity of a particular gene that makes it into a protein and has a function can occur here, because if these proteins that are regulating the translation are not there, then nothing gets translated, right? So you can you can shut off translation of a whole suite of genes that are already transcribed at this level by simply not providing access to these regions that are required, or for the cell not to have these proteins that are required simply to start the process of translation. So that is translational regulation of gene regulation. All right, so and that's really all that I have for eukaryotic transcriptional gene regulation. So now we're going to talk about prokaryotic gene regulation, and that's going to be a little mind-bending. So if you want a little break, maybe now's a good time. Turn this off, come back in a little while, and then listen to the rest of the lecture about the prokaryotic aspects of gene regulation. All right, so if you are ready for more, here's prokaryotic gene regulation. So in your book are a number of figures that I don't like so much. It's confusing. Now, there are some nice examples in the book, and you can use the book for that to see certain examples, nicely colored, and a good way to sort of see what can happen. But there are a lot of ways how genes are regulated, either positively or negatively, and they can have certain regulators that work in one way or the other. It's always binary, it seems. And so I want to put it together in a different way than the book does. I'm using some slides here that a colleague of mine made years ago. I just love them. I think it's just a really nice way of showing this multi-layered way of gene regulation in bacteria, in prokaryotes. So let's start with an important difference in how genes are even ordered in prokaryotes. So unlike eukaryotes, prokaryotes cluster their genes for enzymes in a given pathway. They cluster them together. So if you need 3 enzymes that are required for breaking down a certain sugar, then they're not dispersed through the genome. They're regulated together. Makes a lot of sense. Really strange that eukaryotes haven't adapted that from prokaryotes throughout evolution. There must be something about it that isn't perfect, and that might be that if you lose the regulation of one gene, you lose the regulation of all of them. But what you do have as an advantage is that all of these genes are regulated together. So if suddenly something shows up that needs to be metabolized, all the enzymes that are required for that are regulated at the same time. And that cluster of genes that is regulated as a unit is called an operon. An operon is a set of genes all at this, in one big stretch, regulated in one way. So an operon is transcribed all at once, making one mRNA for multiple genes. And when you have these mRNAs that are now coding for not one protein, but for multiple proteins, that is called a polycistronic message, or polycistronic RNA. Poly means multiple, cistronic means these cistrons is basically a gene, an old word for gene. So if you see that term polycistronic RNA, that means it's an mRNA that consists multiple genes. It's prokaryotic. All right, so it might look like this. You have gene 1, 2, 3, 4, 5. It has one promoter that is regulated by it, but it also has these 2 sequences that are either before or after the promoter. And these are sequences that control the transcription for this gene. So promoter is required, just like in eukaryotic genes. It is the place where the RNA polymerase binds, just like in eukaryotic genes. But we have these 2 types of regulatory sequences that are found upstream or downstream of the promoter. So when you have regulatory proteins that bind here in these particular upstream or downstream regions, that is how you have control. And so this says here, it is the binding of the regulatory protein to the control sequence, this or that regulates transcription, and that binding can either activate transcription, and then we call that positive control, or it can inhibit transcription, and then we call it negative control. And that can be useful if you have genes that are usually on, if you want to turn them off, just temporarily, or genes that are usually off that you temporarily want to turn on. So whether the regulatory protein binds or not is going to depend on the presence of an effector, a molecule that from the environment that indicates to the cell what it needs to do. So now we have either positive or negative control. And now we have effectors that turn on the positive control or the negative control. And we'll discuss all that in much more detail. So we have the terms that you need. We have the operon, we have the promoter, we have these regulatory regions, we have positive or negative control, and we have effectors that affecting these proteins that bind to these regions to determine whether or not positive control of the gene is active or not. All right, we'll come back to all that. So here's an example for positive regulation, and this is from your book again. So in generally speaking, in positive regulation, you have an activator binding a required site for transcription, and the binding site sometimes is referred to as the control element or the activator binding site. So here's the activator binding site. Here's our RNA polymerase. The thing binds, the protein binds, you get activity. Transcription can occur. That's pretty straightforward, right? Now, if the activator is not there, then the RNA polymerase cannot bind. You have no gene expression. That's also pretty straightforward. Activator binding is required for transcription. So that's positive regulation. Now, negative regulation is the other way around. So in negative regulation, the native state of the DNA allows the RNA polymerase complex to be recruited. So normally the RNA polymerase can come on here, and then transcription takes place. So you negatively affect this, you negatively regulate it by saying, well, normally you can transcribe, but if it's regulated, then it cannot transcribe anymore. Transcription cannot occur if this regulator binds. And the regulator in negative regulation is called a repressor. So repressor binding is required for preventing transcription. So the repressor binding site is called the operator. This is the repressor binding site or the operator. Those are the terms for those areas. All right. Now let's talk about the effectors that affect the proteins that bind to determine whether or not positive or negative regulation can occur. See, that's where it becomes sort of complicated. So what role the effector plays depends on whether the operon is inducible or repressible. So that's another binary layer. Is that operon inducible or repressible? I'll tell you what I mean with that. So an inducible operon is normally off. That means it's not transcribed. It's just sitting there waiting to be turned on, to be induced by something from the environment. It's not transcribed unless it's turned on by the presence of the effector. And so this effector will induce the operon to become active. And it's now the question what this effector might be. And we will talk about what these effectors are and where these molecules come from that act as effectors, because that's really the key to understanding the transcriptional or the gene regulation in prokaryotes. So a repressible operon, on the other hand, is normally on, and so you need to repress it when you regulate it. So it is transcribed unless it's turned off by the presence of an effector. So now let's think about what could be some examples. So an operon that makes an enzyme for the synthesis of an amino acid is repressible because it should be on all the time. So it should be something that's normally on because these amino acids are required for the protein for protein synthesis all the time, right? You're making these proteins all the time. They have different functions, but they all need the 20 amino acids, and so these 20 amino acids have to be always be available, and so therefore those genes for amino acid synthesis are always on, unless there's some reason for maybe turning them off temporarily, and so therefore those operons can be repressible. Amino acids are usually required, so we can repress this just when necessary. But another example would be an operon that makes an enzyme to break down an occasionally encountered sugar as a food source is inducible. So maybe a bacterium can use a certain sugar that it normally doesn't get much of, but under certain conditions, maybe some other organism that it lives with or that lives in its environment breaks down a sugar into certain components that it doesn't usually see. But when it does see, this particular bacterial cell has a way to metabolize it and uses that sugar. So an operon that makes the enzyme to break down an occasionally counted sugar is inducible and is only turned on when the cell then actually sees that sugar. So those are the two types of examples. Now, what might be the effectors that determine whether or not we want to repress or induce this gene? So in a repressible operon that makes this amino acid, under what conditions would the cell want to stop the production of these enzymes that are making the amino acids? Well, it might be if there's lots of that amino acid already there. It's made a lot of that amino acid. It only needs so much methionine or alanine. And if you have plenty of that around, why make more until it's needed again? So if plenty of the amino acid was present, then you might as well repress the operon for a while. And so what would be a good effector for a molecule to tell the cell that there's lots of amino acids around? Well, it could be that amino acid, right? It could be that amino acid itself. So you make a lot of that amino acid, you don't need that much amino acid, and so now we need an effector to repress that operon. And what would be the best effector? Well, it would be the feedback of that amino acid itself. And so the amino acid itself is the effector here that then acts as a repressor of that operon. So an inducible operon on the other side that breaks down a sugar, under what conditions would the cell want to start production of these enzymes? Well, of course, if that sugar was present, right? So if that sugar is there, then you want to induce that particular operon to break it down and make energy from it. And so what would be a good effector molecule to tell the cell that the sugar is around? Of course, the sugar itself would be the best way for it to recognize that. And so here we have two examples of effector molecules. This amino acid would affect its own production, and this sugar molecule would affect its own degradation. So now let's put an operon together and see what we would need in terms of all the different parts in order to have a working operon that is sensitive to the environment. So let's use a repressible operon that's under negative control. So an operon has multiple genes and has a promoter, right? So that's the same for all of them. Now, what control element or sequence would this operon have? Well, if it's a repressible operon, it would have an operator, right? Because that is what repressible operons, what a negative control has. So under negative control, we would have an operator in the operon. So that is the region where the binding occurs. What kind of product would these genes make? Well, they would make mRNA. They would make, then amino acids, for example. And these amino acids would then potentially, when there's enough amino acid, we want the regulator protein to bind the operator site, right? We want that transcription now to be shut off. So we need a repressor here, right? We need a repressor, and repressors bind to operators. That's the definition. And what an operator is. It's a DNA strand that or region that binds to the repressor. So we need this repressor, and so this repressor is a protein that is made somewhere else in the genome, but it sits around idly in the cytosol. It's inactive until it becomes activated. And how does it become activated? It becomes activated by that amino acid that now acts as an effector. We have so much of it, we don't use it all. It's around in high quantities. So now it binds to the active to the inactive repressor and makes it an active repressor. That active repressor then can also be, this effector can also be called a co-repressor, and the active repressor can then bind to the operon, where it now acts as a repressor, and then it shuts off the RNA production when it sits there. Now, we need a gene for the repressor that's always expressed, so we have the inactive repressor around so that it can become active when the effector is there to bind, to turn off transcription of the gene. So let's put it together in a way that we can sort of keep these different terms straight, right? I talked about active control, positive control, negative control, inducible and repressible, and we talked about the different types of ways how the gene can be regulated. There can be multiple combinations, right? So I'm putting that here together in such a way that we can see how there's negative control that can be repressible, but also negative control that can be inducible. If we have negative control that's repressible, we need an effector. The effector binds to a regulatory protein and allows the protein to bind to the operator site. So that's what we just talked about. If we had negative control of an inducible gene, then the effector would remove that protein, that regulatory protein, from the operator site. Right? So that would be the inducible would now be negatively controlled. We have positive control by a effector that binds to the regulatory protein and then allows the protein to bind to the control element. So the control element is essentially the operator, but it's the positive, right? We said the operator was downstream of the promoter, and it was always for negative control, and then the DNA strand that's upstream of the promoter that is there if it's there for positive control, and we call that the control element. So in an inducible positive control, we have the regulatory protein binding. We allow that protein to bind to the control element. And in the repressible way, we would have something sitting there all the time that is then removed from that control element. So these are the 4 different ways how you can have multiple interactions and how you can have these different terms explain to you how this gene is regulated. Now, to make it even more confusing, if you will, you can have more than one kind of control for a given operator. So I want to give you an example, and your book has the same example, and you should look it up in the book as well and see, which way makes most sense to you. So the lactose operon, or the lac operon, is an inducible operon that's both under negative and positive control. I want to show you how that is the case. So this gene has multiple, this operon has multiple genes, right? This whole thing is the operon. It has a regulatory region for negative control, the operator. It also has a regulatory gene for positive control. So that is the control region. They're not used both at the same time, right? That's like pushing the brake and the gas pedal at the same time. So if it is inducible, if this is an inducible operon, is it normally on or is it normally off? Well, if it's inducible, it's normally off, right? So it's normally off. You need to induce it somehow to make it come on. If it was normally on, then it would be a repressible operon. So without lactose around, the operon is off due to both control systems. Now I'm going to show you how that is. The repressor is bound at the operator. So there's something bound here, and there's no activator bound to the control element. So this would activate it, but it's not bound. something binding here would repress it that is bound. We'll put that on in a second. And so both of these can be used as a way to determine whether or not we have transcription here. All right, so now we have an active repressor that binds to the operator. Now, we also have an activator, but that's not binding. So both of these are doing the same thing, right? This one represses actively. This one is not activating because it's itself inactive. Now, we could remove this active repressor somehow with an effector, and then that would make room for potential expression. We could also have an effector that activates the activator, and then that would bind here, and that would help. So let's see how that happens. So when should the lac operon be on? When should it be active? Well, under two conditions: when lactose is around, and when no glucose is around. So the reason for that is that the lactose operon will break down lactose, but lactose is really not as good as an energy source as glucose is. Glucose, so glucose is a better one. Lactose is less efficient. So when you have glucose, you might as well use glucose. If you don't have any glucose and you are low on energy, and there's lactose around, well then let's use the lactose. That requires a few extra steps. So essentially you need more energy to put in to break that down, to make some glucose out of the lactose, and then you can get the energy from that. So it's sort of a way for the cell to determine where to put its energy, how to be most efficient with its energy to produce more energy from its food source. So the fact that no glucose is around can be recognized by a molecule that's called CAMP, or cyclic AMP, which builds up when energy is running out. So CAMP is the effector for the activator protein. So in other words, when this molecule builds up in the cell, that's indicating to the cell, ooh, we're running out of energy here. So something needs to be done, and that CAMP molecule will do that. Let's see how it does that. Well, when you have your repressor and you have your inactive activator, when lactose is around, lactose binds to the operator and inactivates this repressor. So it binds to the repressor that sits on the operator, removes it from the operator, and now it allows access for the RNA polymerase to produce these enzymes that could break down lactose. Now the question is, does the cell want to break down lactose, or is that just more work than worth it if there's enough glucose around? So as long as that's not the case, as long as there's plenty of glucose around, there's not very much CAMP around. But when there's a lot of CAMP around it, which was our indicator for low glucose molecules, or for low activity, energy availability in the cell, then CAMP builds up. That then binds to the activator. The activator becomes active and can now bind to the control region. Remember that control region is just that region, the name for a positive regulator, while the operator is the one for the negative regulator. So now it binds and allows now for the transcription of these genes that can then break down the lactose and then make energy from that. So as lactose is now broken down, the effector concentration decreases, right? The lactose itself was the effector for binding to this repressor. Now that decreases at the end. This is even broken down and used for energy. And so this becomes active again. Glucose is also made, and so energy is made, ATP is made, CAMP is decreased. This now doesn't bind much anymore because the CAMP is used up, ATP is made from it, and this becomes inactive. And so here we have our feedback regulation that way, that will then turn off the activity of that particular gene. So Let me sum up what we discussed today. So we had two parts to this lecture, really, the eukaryotic and the prokaryotic way. Genes are regulated, they're not always on. That conserves resources and avoids conflicting messages in the cell. Gene regulation is different in eukaryotes versus prokaryotes. Right? We saw all these various different layers that are a part of the eukaryotic transcription versus the prokaryotic operon model with its negative and positive control and its effectors. Eukaryotic gene regulation occurs on the transcriptional, the post-transcriptional, the translational, and the post-translational level. We have talked about epigenetic regulation that occurs by modification of histones or the DNA. We talked about RNA editing, which changes nucleotides in the mRNA molecule after transcription. We talked about small interfering and microRNAs, which are untranslated RNA molecules that lead to the regulation of other genes. Those were the ones that produce this WISC complex and then either methylate DNA or allow for the degradation of mRNA so that that can't be translated. And then we took a break and we spent some time looking at prokaryotes and how they use operons and how those are multiple genes that are regulated together and how they can be regulated at 3 levels: positive versus negative, inducible versus repressible, and then through the use of these effectors that render the inducers or the repressors active or inactive themselves. And so that all together gives you a good overview of how genomes are regulated both in prokaryotes and in eukaryotes. And with that, we'll leave it at that, and I'll see you in class.